again, and I want to reiterate our NCATS, and it's been a very, very good interaction. And like Veda said, I mean, we're developing at the same time we're implementing a system so that there's a real tight connection between the development of the implementation guide and what the actual system will do. So we're not going to, and it's almost real-time effort. If we find a problem in the implementation guide, we try to bring it into the system right away. And Tyler, and over the next few days, we'll be going into that. So I mean, it's a real tight interaction. And substances and regulatory practice, I mean, they, form the essence of basis of almost every product, so they're real important. They're one of the fundamental levels that we have to organize information at the various agencies. Interactions between substances are responsible for nearly all pharmaceutical activity, and we want captured it. And there are linchpin again in our agency and in most regulatory agencies on how to organize information. Unfortunately, it's about 7 years ago at the FDA, we didn't really have a strong registration or identification system. We just had a bunch of names that we had gotten in applications, and we link it by the names, and it wasn't a very satisfying effort. Company codes change all the time. Companies get bought and sold. Full names for a given chemical. You can have 300 names that could be legitimate, and so we really wanted to have a system where we can seriously identify what the substances were and then assign a code, just single code to it, so we'd be able to trace things across products and across applications. Again, we have to do that. We took it as a scientific approach. What are the scientific elements necessary for defining substances? And 1238, and we're already pretty far along with the substance registration system that Frank and I have been working on in Yulia for a number of years. So it really helped us in sort of learning the standard and developing the standard. And this is the slide that Beta showed you. And again, at the pharmaceutical product level, and this again could be a global ID as well. Basically the substances will be in there, and what we're calling specified substance group 1 may be in there as well, and that would be multiple ingredient, most substance ingredients like a lot of the pigments and colorants and some of the flavoring. So the old model, it's a substance. So again, from us, the substance is a molecule or something like that, but we really had to go beyond that because in the regulatory world, we FDA. We're not only drugs. I mean, food is first, really, and it was really where the agency started in many ways. So, we really want to say what's a substance. So now, it's something that has matter and capable of sort of separate existence. So, salmon has matter and capable of separate existence. So that to us would be a substance. So it's not only the molecular level, but it could go all the way up to an organism, anywhere from an atom. We're defining atomic particles yet, but maybe electrons, but again, anywhere from an atom all the way up to an organism could be a substance. So, and this is Frank and I trying to, and in dirt, we do have dirt in the SRS in college, and we were arguing about how we should define it. But we came up with a, again, it depends what house it is, and but again, that's what a substance is. And again, we define a substance based on what something is, and for the most part, now it's made or how it's used. So recombinant salmon calcitonin would be the same as the synthetic calcitonin that you made in chemical synthesis, or if you isolated it from a salmon, it would be the same substance. It made different impurities, different specifications, but the essence of it is the same substance. So we try to define a substance based on immutable properties, and it's independent of physical form, grade, or purity. So water is a substance, whether it's ice or steam or some other form or liquid, and those different levels that we'll handle at the specified substance thing. But that proteins, pretty much their sequence and type of glycosylation and complex materials and biological matrices are defined by a variety of ways, either taxonomic or anatomical or fragmentation information or the substances that are present there. At the IDMP, we came up with 5 groups of elements to to define substances. So chemicals is one group, and they're defined primarily by their molecular structure, which is activity in complete stereochemistry, if we can get that. Proteins, again, by their amino acid sequence and type of glycosylation, and then modifications. We're not getting into microheterogeneity at the protein level, so we made it into it at the specified substance group 1 level to try to distinguish some of the product that may or may not be biosimilars. But again, we're doing it at substance level. Again, if it's, and again, our slice of glycosylation was broad in that it was just mammalian, human. There's a list, and probably we'll go over it later when he does a protein demonstration. Nucleic acids are defined somewhat similar to proteins, and again, it's sequence, type of sugar, and linkage, and modification. And for these two things, there was a separate effort that, we sort of did this independently, helm effort, and we pretty much are doing things the same way. And again, we may talk about it later, but we will probably, try to merge those efforts so there's a single way to define proteins and nucleic acids. Polymers in many ways is our most difficult thing to describe, to distinguish. Again, it's structural repeat units, the type geometry, the type of copolymer, block or random, racial monomers, and then, some indication of its molecular weight and its molecular weight distribution. And again, a lot of times that's physical properties. There could be other physical properties too that could be defining. And structurally diverse substances are ones that don't fall into the previous 4, and that's your cells, your viruses, your things like that, and it's mostly taxonomic information, physical modifications, physical properties, and things like that. So those are the 5 elements, and every substance will fall into at least one of those elements, and it will be defined based on one. So if you have a polymer modified protein, we predominantly define it as a protein that's been modified by a polymer. And you're trying to fit everything into one. Again, the good thing about the NCATS system is that we can have multiple definitions. So we could also define it as a polymer modified by a protein instead of a protein modified by a polymer. But it will have the ability to capture that sort of information, which is unique in a lot of these chemical registration systems that will be able to capture multiple definitions. For a lot of chemicals, you draw multiple structures that most chemical information systems would think they're different. And so the ability to capture multiple definitions, we think, is really important for limiting the ambiguity of substances. And the need for further specification. Again, we really need the time material to manufacture and process, and that's kind of group 2 level. And we haven't addressed that yet, and that will be something we'll address in the future. We need to tie it to specific grade, and that's level 3. Specified substance, we need to obtain specification information, to have a good specification database so that, if the same substance is made by multiple manufacturers, you have a similar specification range. I know when I worked at USP, we were always trying to figure out what was the real specification. It can vary if the methods of synthesis is different. They have a good specification database, so, we're limiting a substance in one monograph, they're similar throughout. And there's a need to obtain information on processing materials and start materials, and a need to establish and monitor the supply chain. So that was the need for the specified substance. And it's going to be implemented with the GINAS application. Group 1 has already been implemented, and again, that would be where we would distinguish multiple substance materials. Cytocones, a good example. Physical form, we distinguish steam from wet water, extracts, things like that. Even in Europe, even oils and things like that would be group 1. The manufacturer, who manufactured it's important for us to know for inspection purposes and a lot of other purposes. And then minimal manufacturing information would be group. If somebody, say, went from with a recombinant technique to synthesis, that may get a new group, even though the manufacturer stays the same. And then the need to get what the grade of material is, and again, that would be working with USP and EP, really get that in and with their specifications and sort of follow that so that we're able to do that. And then group 4, which will be the last thing that we implement, will again have detailed manufacturing information and where we go starting materials, processing materials, the very detailed manufacturing information. That's in the CMC section. So that will be something we'll in probably next couple of years we'll start to implement within GINAS, and with the analytical data and things like that. So that's the supplied substance, and there's really a need for a global database. It's a global marketplace for ingredients. I mean, Pentagonis and I were in the dollar store yesterday, and we things are coming from all over the world, and they may have been in India or Pakistan in one day, and they're in the U.S. the next day. Hopefully they've been tested and are good, but again, we need to know what's there, and we think a global database. And in many ways, it's kind of a tragedy is that the method of manufacturing is probably never better, but anyways, throughout the world, the quality of drugs may not be the best. I don't think it's the worst of all time, but it's certainly in African countries and in other countries in Asia, the quality of drugs sometimes isn't where it's at, and there's a lot of company, a lot of other stuff going on in those countries, and I used to get a handle on it. I mean, one time I bought coins that were supposed to be real Roman coins, and there's a database and they weren't, they were better fakes, but I went on a database and found out they weren't real Roman coins. But there's no database now where we can say, where are the counterfeit drugs at least that I'm not aware of, what do they look like, and what are they doing? And I think that's real important. And then this, may be able to feed into that in some way. And again, 11238 was recently developed to define that it's easily extendable to other things like dietary supplements. In many other countries are drugs. In the U.S., they're regulated by foods. Cosmetics, a lot of them now in the U.S. are becoming drugs because they have a sunscreen element in it. And so there's a lot of need. There's no single definition of what a drug is throughout the world. So we have to really handle a lot of different things. We went to this GINAS effort was that we realized 11238 is a complex standard, and to implement a system based on it on an individual basis could prevent the adoption of a standard. There's a lot of standards that get developed, they never implement it. I mean, 870 has a huge list, even ISO ones and things like that have never been implemented because there was no system. And we thought it's very important to have a system that everybody can use and to implement the standard. And again, we think safety analysis based on substances with global data will be much better, and UMC does a lot of that, and a lot of trying to map things or figure out what was really given to the patient, and having a global system will allow that to happen a lot better. Again, better coordination of regulatory activity in clinical trials and all that. If we know substances, where they're made, and, we're probably able to alleviate drug shortages. A lot of times, when the enemy hits a certain area, a lot of times there's one plant that makes most of the things. I was in Germany, and they're telling me there's one plant that makes the bulk of levothyroxine, and if a guy disappears or, something happens in his plant that could really create a big problem. And so at least to realize where there's vulnerabilities in the drug supply system and maybe trying to address them before they actually become problems would be really good. And we think that standards and specifications can converge more rapidly.
So are you differentiating between global software and global database? So will GINAS be just the software?
Yeah, there will be GINAS will be, GINAS is the organization, and again, we haven't really formed it yet, but that will be a repository, mostly of public information, but it may be an organization that you can go through and register your substances to different regulatory agencies throughout the world. That hasn't been put out yet. The software will be something that we're going to distribute, prior to GINAS. And that's, again, they're not a software development. They do a lot of software development, but eventually they're doing this, getting it off the ground, getting a good system going. Somebody else will take it over in several years, and that will be what the organization is, and it may also be a operation. But again, we're still working it out. We haven't done anything yet. But it would be something that either regulators trying to kind of share the information, we haven't worked that out, to be honest with you. Yeah, but we'll be distributed for the next several years. We plan to have a production version of the software we're distributing now isn't a production version, but by July we will have a production version that at least at the FDA we're going to be using it for production, and that will distribute, and we'll probably have a workshop at that time to really, things out and things like that.
So the software is GSIS? And
yeah, the software, yes, will be GSIS.
The content.
Yeah, right, and that's something that NCATS will distribute, and probably maintain it for the next several years.
Yeah, we should probably make the comment that the name for the software, for the equal software component of it, has gone back and forth in time.
Yeah.
GSIS, everyone will understand what you're saying. You should say that, but we've also added a couple other things. Some of them we can't repeat, but that's we understand that there's always that ambiguity there, and that can cause a little bit of confusion. If you just call it the software, then that will also be the best.
The whole idea, this is you go to one archive system, and we have as much information as we can put in there for various sources. Some we would maintain, some we link out to other sources, but it's just would be a one-stop shop for substances that are of interest to the to the pharmaceutical industry and to the Spectra. We'd have anything we can make release, we would release. We're working closely with USP and a lot of data on a lot of substances, and maybe to get in and stuff like that would be very good.
Sorry, this is USP clinic.
United Pharmacopoeia.
Okay.
This is what USP is, and they've been involved in the SRS for almost since the beginning as well. The substance registration system. And there's our vision, and again, you have to either produce guidelines or regulations, but we think that every substance should eventually be registered prior to every submission, so that all you do is put in your submission, and again, the ECDD may be better organized, especially the C section, that before you submit something, unis on the stuff, so that we're able to get in the databases easier, do evaluations a little bit easier. And then, the whole idea of this is we'd have a unique identifier that would be permanently associated with each registered substance. Sort of and again, we're calling them unis at the FDA, or maybe their name that's developed. And then the unis will be used to develop meaningful relationships between substances, submissions, a lot of other stuff. And that's what we've sort of done at the FDA. And, again, we want to expand it. I mean, I know companies are usually much further along with stuff. And then we use an SPL-like process to obtain and exchange this information related to substances. And then we link them through link both within and outside the FDA, like USP in their dictionary. In the USP dictionary, they have unique codes. And we work with PubChem and with the Special Information Services quite a bit, and there's a lot of unis in both of those things. Again, the EA we're working with, and we're mapping their identifiers to our identifiers a bit. We work with the Personal Care Products Console, which is cosmetics, and Stan is part of that, and we, they put unis in a lot of their publications. Martindale, we worked a lot in the past, not so much recently since they changed the editor. The Merck Index, and then Wikipedia, we put unis on Wikipedia to help find information. Drugs we work with, and we're big things, and we give them unis. I don't think you guys have put them forward yet, though, Melon, right? Or,
No, we've been running some.
Yeah, but still, mapping, and we go into our system, you'll see that we indicate that that's a substance name for WHODD in our system. I think Frank released those names on our NL, so you can tell that the one mapped to WHO drugs that we mapped to them.
There's over 20,000 units
that WHO drugs, yeah. And 1,000 to the EPMD. It is, there's different, even with us. I mean, we had a generic polyethylene glycol uni. We don't like that. We want to really get specificity with unis, and a lot of times the terms that people use aren't that specific. They map to a number of compounds.
And actually, most of the ones that we're not able to map yet in the May list, EPMD, are things where it's mostly homeopathic, and every separate homeopathic strain has a separate code. Yeah. That would be a group 1 or
over the summer, we'll implement it and link a lot more codes, hopefully. That's our vision. The history, again, this grew out of a meeting hosted by USP and NCATS at USP in February 2013. The meeting was hosted by the CBG in the Netherlands in September 2013. We had a third meeting hosted at USP in June. Again, we're using software developed. We released a version, and we worked with Health Canada. GINAS prototype has been up there for almost a year and a half, so people could look at it. They can do mock registrations. They can put in there. Again, it's and I've done that a few times. Again, it's not. We have it just to make sure it works. And we worked very closely with a number of regulators: Canadian, Dutch, German, Swiss, and U.S. regulators. EDQM and USP have also been involved in the development of the system and standard. Again, the idea is that the software, you can deploy it locally. We'll show you can deploy it on your own personal computer. You have all the data. I mean, it's not a huge, we're not trying to write the biggest database, but we want to get the stuff that's important in there. So you can put it on your computer and run locally here. You can register it. Eventually, our goal would be a button that, sends information to FDA, send it to EMA, send it to Dutch, any of the regulatory agencies that you want. And then, we have to find out some secure way of sending data for companies, but it's something that companies can keep behind their own firewall and then, just the information when they think it's appropriate. Okay. Again, data integration, I mean, that's, substances are nice, but unless you really integrate with other information, it doesn't mean that, and then this is what we think actually where we'll be. And I think working with NCATS has a lot of ADMIR data that they map to and stuff. And in many ways, the FDA has the most expensive or valuable data in the world. But again, if you try to find it, sometimes it's not that easy, and it's not organized in a way. If you look at even our reviews, if you go on drugs at FDA and look at the Clean Farm reviews, there really is a treasure trove of information there that can really help, we think, drug development, and NCATS thinks that as well, and so we're going to try to get that better organized so that we'll promote drug development in the future and things like that. So I know I was involved in drug development for many years, and you want things to fail early, or you don't want to have things fail in clinical trials necessarily. You want them to fail before that, and you certainly don't want them to fail after they've been approved, because then your company's at risk. And so again, trying to get the information. Again, it's always hard. A lot of drugs are very close, whether they should be approved or not be approved, and it's always a difficult decision to make sometimes. And again, that we hope the system will do by integrating data. Again, they will be entered through an API for the most part. It's predominantly open source. It will be web-based. It will comply with the 11238 substance model, and it will be freely distributable to regulators, other regulators, and companies. It's database neutral. You can run it on Oracle. You can run it on MySQL. I don't know if you tried Postgres. I don't know if you tried here, but again, H2. It comes with H2 currently. It will work with a number of web application servers. It's Java-based. Again, a RESTful interface, and there's a lot of leucine indexing involved in the system. And again, Tyler and Trung will go over it in much more detail. It's like Beta's slide, again, it would be an EMA if we come up with a common system, but again, that would be the basic idea, is that there'd be a public base, and again, that's currently up at Health Canada, and it may stay there where we put public information, and then everybody can then send it to the thing. And the progress so far, the prototype's been deployed at Health Canada. The first version of modules was distributed earlier at the previous meeting. The system is being deployed within the FDA environment. NCATS, again, provided a June meeting. The second version will distribute today. And again, it's not a final version. We're really looking for a lot of comments, and we've migrated much of public data into the system that FDA had, and NCATS will probably be adding data as well. Again, this is just some stuff that would be important, and most of the stuff we have open, and the site, the Tribot site that NCATS hosts, will have all the presentations, and they're developing a lot of documentation right now too Next meeting will be held in the summer of 2015, perhaps Washington, perhaps at USP, because they actually provide food and coffee. Again, we haven't got a commitment there yet, but we would certainly appreciate that. And then there may be another meeting in Switzerland with the ISO thing later, and so that's where it's at. Here's the most important side, I guess, is this has been really a global effort with a lot of people at the FDA. A lot of this started with Randy Levin and SPL, the need for substance identifiers, and Mitch is a contractor who's been working with it. Our developer we have one developer on our SRS system, and she's done a fantastic job. Archana Noasha and Frank and Amelia are FDA employees that sort of enter data occasionally and do a lot of stuff on that. The NCATS team, D'Amica used to work with us. Heather used to work with us. We lost the best people, and then Trung has really developed this thing, and their team leader, and Noel has been leading the effort at NCATS. We've had a strong participation from a number of foreign regulatory agencies. Thomas Balzer from BfArM. Hermetric has really been a lot of moving force in Europe to bring this thing forward. We've worked with Japanese quite a bit, with Takesha Levis here. Philip Wireman couldn't get here, and we're working with Kew Gardens. Kew Gardens is going to supply all of the technology for plant-based materials and things like that, and we're going to integrate that closely with them. And again, we've worked also with others. With WHO should have been on here. I don't know if I missed it. I mean, again, it was a limited slide, but Madeleine's in a lot of meetings. And then the TJ Beta and Marion Splack are involved in the thing. And like I said, Sabine Broach is really the one who started this effort for many years, and Paolo and Ilaria and kind of, I don't know if I got your names mixed up here, man. I don't know which comes first, but and then we've had a lot of interaction with industry. Andrew Marr has been involved from the very beginning as well, and Michael Triar. And Paul Houston was the original project manager for the IDMP, and he was really responsible for bringing it forward in many ways. And Claude was involved with the Chemist Jarvis, and then the excipient industry has been very proactive in this. They really want to see this happen because, again, they are registering excipients all over the place, and they've been really pushing it. And Dave Schonecker and Catherine Ullman have been involved in that. And then at USP, Fuad Atuf, Patrick, Kellen is on the phone, was involved in helping write the herbal extract, and so that's again the